Hello everyone, and thank you so much for joining me for today's lecture on the nanotechnology. So we're going to talk about the nanomaterials, talking about the nanotechnology. We're going to talk about nanomedicine, and of course talking about the human health. That's because we need to use the existing technologies and to develop, of course, the existing ones as well, in order to find better, safer, and products that are more efficiency for animals, of course, but here, for the sake of this lecture, we talk about human health. So what we are going to talk about today? We are going to have an introduction related to the nanotechnology. Of course, you may already know what is nano. This is 10 to the power of minus 9 meters. So we are talking about very minuscule and very small I would say molecules or even instruments that we can make. Of course, because of the physical and physico-chemical properties of elements that we have, we may not be able to make, for example, ultra extra small engines. This is what you see in different science fiction movies. But we can always look into different properties of different metals, for example, or different molecules in order to use them in medicine. Now, the use may be obviously being served as a vehicle for a drug. We can, for example, again, use drugs that we are going to control the movement in the body. This is the idea as well. But we'll also use nanotechnology and the products for surgery. We are also going to look into some examples. And overall, I just want to let you know that when we talk about nanotechnology products, we are talking about medical devices and obviously combination products where we have a device and of course a nano device in this case, and we are also going to have molecules of a drug, and we are going to use that technology. We can also visualize cancer cells. We use, again, as I already mentioned, the physico-chemical properties of metals as well in order to be able to look into a cell. We are going to talk about, again, the quantum dots, where we can use those instruments as well, nano instruments, in order to get into the cells and then to visualize some structures. So this type of nanotechnology allows us also to visualize, again, anomalies in cells as well. So we will then go over what is, again, nanotechnology. This is already what I talked to you about. Then we are going to cover, I'm going to go through some examples of nanomedicine. Obviously, because of the fact that lectures are not always 6-7 hours long, I already selected a few examples, but at the end, I'm going to give you a few slides about the potential applications of the nanoparticles in medicine, and I'm going to talk about the future of nanotechnology. Obviously, we may not know always what's coming in the next years, but at least the philosophy behind research and the philosophy behind, of course, I would say medical interventions. And this is why, again, we are talking about nanotechnology. So what is nano? I'm going to read the slide for you. Nanotechnology is the understanding and control of matter at dimensions between approximately 1 and 100 nanometers, where unique phenomena enable novel applications. Novel for new. Dimensions between approximately 1 and 100 nanometers are known as the nanoscale. Now, unusual physical, chemical, and biological proteins can emerge in materials at the nanoscale. So as you see, because of the chemical, physical, and biological properties, we are limited, okay? We are limited because we cannot, for example, make molecules that are smaller than some size. That's why the nanotechnology for now, obviously, this is the limit of what we can do. Obviously, in the future, we don't know what is going to be the technology. If we can make matter, for example, matters, even molecules that are going to be smaller, or atoms, we don't know. But for now, we are limited to the nano size. Now, these properties may differ in important ways from the properties of bulk materials and single atoms or molecules. Now, this picture is really showing you what is nano. So on the left, you have a butterfly, and then you do have, again, this is again I would say this is a bigger, or a picture in much more detail of the wing of this butterfly. But when we are looking into the nanotechnology, here we are looking into again 1 nanometer resolution, and this is what you see. Sorry, it's a 1 micrometer. This is on the right side. Now, when we talk about nano, this means that we have to, we will have a resolution of 1,000 micrometers, sorry, not 1,000, 1,000 times more, I would say, higher resolution of the picture that you can see on the right. So it's very small. Now, engineered nanomaterials now, what are these? It means that they are any intentionally produced material that has 1 or more dimensions of the order of 100 nanometers or less, up to or down to 1 nanometer, or is composed of discrete functional parts, either internally or at the surface, many of which have 1 or more dimensions of the order of 100 nanometers or less, including structures, agglomerates, or aggregates, which may have a size above the order of 100 nanometers but retain properties that are characteristic to the nanoscale. So when we talk about nanoscale, here we are talking about cells, about the structure of the cell membrane as well. We are looking, and we are able, of course, to make nanomolecules, even nanorobots as well. These are molecules that can do something. They can, for example, adsorb against the cell membrane in order to allow us to, look into the surface of a cell, for example. They can also penetrate the cell through endocytosis, and then inside the cell, we can, for example, make vesicles that are going to have some, cell membrane around them. Once they are, within the cytoplasm of a cell, or cells with some disease, they are now going to release what is in their content. Now, the content may be, for example, gold, I would say some crystals, for example, or it can be gold, very small pieces, nano pieces of gold as well. It can be any other type of material that once we are using, for example, ultraviolet or even light, we will be able to see where those nanomolecules are adsorbed, and we may now be able to see the surface of a cell, for example, with a cancer, or we may now look into some intracellular structures, if, of course, the structure of our nanomaterials will allow us to visualize. So this is one of the uses of nanotechnology. But we're going to look into much more, applications of nanotechnology. Now, we also have another term, which is engineered nanomaterials. So you may see in different books or different articles the abbreviation ENM. This is any material that is deliberately created, such that it is composed of discrete functional and structural parts, either internally or at the surface, many of which will have 1 or more dimensions of the order or of course 100 nanometers or less, so down to 1 nanometers. Now, these are the applications of nanotechnology. Obviously, and as I told you at the beginning of this lecture, we will not have time to go over each and every of these applications, but allow me to start with the middle of this picture. We talk about nanoparticles. So when we talk about nanoparticles, these are particles at the size of 100 micrometers to, of course, down to 10 to the to the power of minus 9 meters. So here we can have nanoparticles that we are using in textile. So these are medical textile. And again, we can, for example, have anti-stain textiles. We can have electroconducting textile. We can have synthetic polymer hybrid fibers that are going to be used as textiles. Now again, why? We are using them, for example, for skin, but it's not just external. It can also be in internal tissues. We can also use nanoparticles in the biomedical field, meaning that we can use nanoparticles that are going to, be attracted, I would say, to cancer cells. We can use them in cancer therapy. We can use them as biomarkers. But we can also use them as drug delivery systems. And I already talked about the insulin. So for example, for insulin, we can have nanovesicles at different speed of degradation for nanovesicles containing the insulin, where when they are injected in the body, those vesicles are going to gradually degrade, and then they are going to release gradually the insulin that exists within those vesicles. So it's an excellent tool, and it will be, of course, an excellent tool for drug delivery as well. We also look into, it's already done, of course, but we are still looking into developing more of the nanoparticles where we can make them like they are sensitive to specific cells. But here, of course, we are going to use different types of molecules, such as, again, immunoglobulins. In this case, we can make immunoglobulins that are selectively attracted to cancer cells, a specific cancer, and then we are going to use those, attaching to those immunoglobulins, nanoparticles that contain an anti-cancer drug. And in this case, when we are using or injecting, for example, the nanoparticles with those immunoglobulins, we will see that, and we observe that, it's already done, but we can see that those nanoparticles are going to be, again, attached or attracted to the only cancer cells. And then they are going to obviously release their content, and then to do the, the job, I would say, what they have to do. We can also use nanoparticles in healthcare. We can use nanoparticles for, again, being agents for, I would say, for killing bacteria. Or, for example, ultraviolet protection. We can also use nanoparticles in food and in agriculture. So we can make nanoparticles that are going to selectively, again, attack different fungi. And again, they can have, again, some products that are going to kill specifically the fungi and not, of course, the plant. We can also look into the use of nanoparticles in the industry. So here we can use them as industrial catalysts when we are making, again, we are doing some chemical reactions. We can use nanoparticles in electronics. So, for example, the electronics, the computers that you have or the hard drive that you have, they are all having nanoparticles. Of course, here I'm not talking about the use of nanoparticle in medicine, which is the title of the presentation. But since we are talking about nanoparticles, allow me to completely, I would say, go over the complete explanation of how we can use nanoparticles. So for the electronics as well, we can, for example, have high density data storage. Why? That's because, for example, your hard drives, they have, again, a layer of plastic, and over that you have some ferromagnetic vesicles, for example, and higher is the resolution, so lower is the diameter of the nanoparticles on, again, on those disks. Higher will be the, I would say, the not the size, but I would say the capacity of that hard drive, for example, to keep the data. Okay, and again, the data, that's because again, I'm going to invite you to, of course, look into some information about the hard drives as well, because what happens is in the hard drives, we are going to magnetize the particles, and when they are going to have a specific, I would say, magnetic pole, no plus or minus, they are going to be used as a 0, 1 or 1, 0, which is the code, the binary code. And based on the binary code, we will be able to store data on the hard drive and then to read the data from the hard drive. So everything is based on the use of those nanoparticles as well. We can also use nanoparticles in the environmental, of course, sciences. So we can use them as environmental catalysts. We use, for example, here, of course, some, for example, carbonoclastic particles. We can add some bacteria that are carbonoclastic. It means that you're going to degrade carbon, and here this is for using those particles in order to, I would say, degrade faster the oil that it may be spilled from a tanker in the ocean. So we can also, of course, use the nanoparticles, as you see, for polluants and scavengers, or for the pollution, we can have monitoring sensors as well. So it's very interesting to see how the nanotechnology is useful, okay, it's useful, and again, we need to know about the physical, chemical, and biological properties of different, I would say, particles or nanoparticles. And obviously, the last one is about the renewable energy. So the nanoparticles allow us to, again, increase the hydrogen production, and because they can be photocatalysts, they can also, of course, be used as a fuel cell catalysts or fuel additive catalyst as well. So I don't want to go, again, outside of a topic, but as I already mentioned, this is a very interesting topic, and this is the future as well. This is where we go, because we are going to see how we are going to use the nanotechnology in medicine, and it's very promising, extremely. Now, coming back again to the size, because I want to make sure that we understand that very well. Nanotechnology is the understanding and control of matter at dimensions between 1 to 100 nanometers, where unique phenomena enable novel applications. So now on the right side of the slide, and this is a picture from National Cancer Institute, you can now see that we are having again on the left side of the again the picture, the nanometers or the size, and we see that, for example, the size of a water molecule is 10 to the power of minus 1, and then we have nano devices now, which is which is the nano device. We have 10 to the power of 2. This is again the this is 100 nanometers. So as you see, we are talking about very, very small structures. This is where on the line you see 1, 10, and 10 to the power of 2, or 100. So here we are talking about different molecules, different vesicles, and of course the assembly of different vesicles. And now if you would like to compare even 1 nanometer or even 10 nanometers with the size of the white blood cell, this is what you see. So when you see 10,000 nanometers, you see the size of the white blood cell. So we understand very well that by using, we are able, of course, to use the nanovesicles as well, because they are up to 10,000 times smaller than the diameter of a white blood cell. Obviously, white blood cell is also a big cell, but usually our cells are between, again, I would say 1,000 and 10,000 nanometers. So we can use, of course, those nano devices. Now you can see, for example, the nano devices. You have nanopores, you have dendrimers, you have nanotubes, and you have quantum dots and nanoshells. We're going to look into what are the quantum dots, for example. We use them in order to do the diagnostics, and then to characterize or to explore the surfaces of cells. We are going to use them because these agents or these nanoparticles, they have different properties that will allow us to use them in medicine as well. And obviously, if you put, again, a dot on your paper, as you can see here, if you see the white blood paper, this is 10,000 nanometers. Now, if you have 100,000 nanometers, 1 million nanometers, this is the size of a dot that you are going to put on a piece of paper. So you see how a nanometer is small. Now, let's go over the nanotechnology. What is nanotechnology? So the first, we can talk about the passive nanostructures. But don't forget that we also have active nanostructures, we have systems of nanosystems, and we have molecular nanosystems. So let's go over the evolution. Okay, on the left side, you can see the timelines. Okay, so for the passive nanostructures, for example, we are talking about the year 2000, and here we are talking about the first generation products. When we are looking into the second type, I would say, or active nanostructures, we are talking about approximately years 2000 to 2005. Then we were again seeing some evolution, more evolution in the nanotechnology. That was around 2010 when we were using systems of nanosystems, and obviously nowadays we are looking into molecular nanosystems. Now, for giving some examples, in the year 2000, talking about the passive nanostructures, we can give you the examples of aerosols or colloids. We can give you examples of coatings and with nanoparticles. So here, as I already mentioned, we were using coatings as well, but the best example is the extended release insulin, where we are having, again, composites, we are having vesicles being made by polymers, and those polymers may have different required time in order to be degraded and to release insulin into the body, and that's why we have those extended release, I would say, injections. But we also, again, have other structures, okay, so like ceramics, and we use those passive nanostructures in coating of different drugs as well. Now, when we are talking about the second, I would say, level of evolution in nanotechnology, we are now in years between 2000 and 2005, where we are talking about active nanostructures. So we use them, of course. Here we talk about bioactive and health effects. For example, we have targeted drugs and we have biodevices. Then we also talked about physicochemical, chemically active nanostructures like 3D transistors, amplifiers, actuators, or adaptive structures. So these are all, again, structures we started, for example, as you can see at the left of the rectangle in, I would say, blue. Yes, it's blue. Second, you see that, for example, we were able to produce prototypes of very small molecules. Then around 2010, we are talking about systems of nanosystems like guided assembling. These are 3D networking and new hierarchical architectures. We think, or we talk about robotics, and of course we talk about evolutionary, I would say, nanosystems. These are ideas, obviously, here. We do have prototypes as well, but here we started to look into not only the technology, but also the risks and benefits, in addition to ethics related to the use of nanotechnology, because we also use nanotechnology in vaccines. We also use nanotechnology in vaccines where we are using RNA, double-stranded, of course, RNA as well. So we are going to cover that as well. And then for the last part of this slide, we also can talk about molecular nanosystems like molecular devices by design. These are, again, atomic design, and obviously they can have emerging functions. Now, let's go over the application of nanotechnology. We already went over that, but here I would like now to review this, knowing that we already have nanotechnology tools, and we already have, many even drugs and combination drugs that will help us to, use those that technology in order to, again, give or increase the quality of life of people. So we already talked about the application in energy, okay? In medicine and drugs, that's why we are here, and we already talked about combination products and about the medical devices. But we also have nano-biotechnologies. We have nano devices that we are going to have, I think, one example. We can also use nanotechnology in optical engineering as well, in defense and security, bioengineering, cosmetics, and nanofabrics. But the main thing is that because of the size of these vesicles or these particles, when we are having a specific physical or chemical or biological activities associated with the structure or the, I would say, inner structure of these particles, we can now look into how we can use them in medicine. This is why, again, we are here today. Now, let's talk about nanomedicine then. This is a nanotechnology applied medically. Now, we have new breakthroughs in medicine, for example, advanced biomedical research tools. We can, for example, do the study of DNA and its component genes. We can we already developed, of course, diagnostic tests. And, of course, we can use them in bone implants, as well. And, of course, we have so many other uses for the nanotechnology. So now, don't forget that when we talk about nanotechnology, we are talking about molecules. It may be molecules that are they are already made, so they are biological products, but they are having a very small size. So here I gave you the example of immunoglobulins, for example. So you don't need to necessarily create your nanoparticles in the lab. You can use biology. So these are biological products that, and you can use bacteria or cells in order to produce your nanoparticles or to produce your, for example, double-stranded RNAs. We can also use existing living organisms in order to I would say, to work like a supplier for us for nanoparticles. And then we are now going to assemble those nanoparticles in a lab in order to reach our therapeutic goals as well. So when I'm talking about nanoparticles or nanotechnology, this is not just a piece of metal, for example, that we are thinking that we are going to make and then it's going to act as a robot. No, we are looking into even assembly of different types of again, molecules or sometimes the devices, it's very small one, minuscule ones, and the assembly can be based on any biologically, I would say, biological product, or it can be, for example, a very small, very, I would say, at the level of a nano structure of a spring, for example. Not a long time ago, I was looking into very small, again, at the level of the nano size springs that were used in some type of therapy. We also have, for example, therapy with magnets. We are going to go over that as well. Now, one of the very important part of nanotechnology use is the drug delivery methods. So these are systems that deliver drugs to specific sites. Now, the sample methods these are, we call them smart drugs. We can also make nano-composite hydrogel systems. I'm sure that you heard about Japanese scientist that was, again, in last week, it means in February 2025, where they were talking about nanogels that were made that would be able to make cancer cells and push them back to the stage of cancer stem cells. So we are using this technology, and of course scientists all over the world, they are successful in finding new drug delivery methods as well. And of course we can talk about magnetic nanoparticles, because when we are dealing with again magnetism, we can also affect some physiological activities as well, and this is again one of the examples that I'm going to give you shortly. So let's talk about the smart drugs. We talked about immunoglobulins. So here I'm talking about antibodies, okay? And here, for example, what we need to have the smart drugs is first to think about, attacking a specific antigen. So when we characterize the antigen, we can have or make the antibody. And when we are producing the antibody, we can also create vesicles, nanovesicles, and then to attach the nanovesicles to the antibody that will then be released in the blood, and then, or of course in the tissue, and then hopefully this is going to attack all of the antigens that are present on the cell membrane. So here, when we talk about smart drugs, we talk about attack-specific antigens. We are talking about immunotoxins that are protein in nature that we can make, and it consists in an antibody part and toxic part. So as you see, the structure Y that you see, we are going to add into the antibody the toxin. So we are going to have an immunotoxin antibody and the toxin. And then we are going to, of course, have the antibody part that will attach to a protein on the cell membrane. And once the cell membrane is now, of course, it's already attached to the antibody, now the antibody can or release the toxin in the proximity of the cell, or it can undergo the endocytosis, and then the toxin attached to the antibody is going to be released in the cytoplasm, and it's going to selectively destroy those cells. Now all we need is just to know, of course, what is the target cell. We need to do for example, if that's a cancer, we are going to get the cancer tissue, we are going to do a cell culture. Now we are going to see what are the particular antigens that exist on the, again, or even any protein, specific protein that is existing on the cell membrane of the cancer cells. And then we are going to create an antibody that is going to recognize only that specific molecule, which can only be found on the cell membrane of a cancer cell. And then we will just use the very natural process here, which is endocytosis, in order to kill specifically and selectively those cancer cells. Now, nano-composite hydrogel system, these are thermotherapeutic process. They release, of course, drugs that are encapsulated on heating. This is, of course, the gold nanoshells or nanoparticles, and they are, of course, going to allow us to see them with the ideal wavelengths of light, of course, that are infrared, but at the same time, we can activate them with that wavelength. Now, you may say this is very interesting, but if, or what if the cells are deep and we cannot, again, use those wavelengths? You're right. We're going to have a slide at the end of the presentation as well today, where we are going to look into some limitations of nanotechnology. So when we are now talking about light activated nanoparticles, or like for example here nanocomposite hydrogel systems, we know that of course in addition to all of the pros or all of the benefits, we also have some limitations for the use of this type of drug delivery method. Now let's talk about the magnetic nanoparticles. So here, as you can see in the picture on the right side of the slide, you have drugs that are bound to magnetic nanoparticles. Now they carry drugs to malignant sites with magnetic fields. And they will release the drugs by enzymatic activity, because this is again what is attached into again those nanoparticles. Now for this, I'm also going to invite you to look into another video that you can find easily on even on YouTube, where we are talking about magnetic nanoparticles, and you're going to find several movies where you have a physician that is of course using the magnetic nanoparticles. And since the physician wants that those magnetic nanoparticles with the drug attached to them go to a specific, for example, organ or a place, they place a magnet on for example, the tissue. For example, one of the movies that I watched was a physician using magnetic nanoparticles, and injected, of course, and then they were using a magnet that was, I would say, kept against the knee of the patient, because the physician wanted the magnetic nanoparticles to go toward the knee of that patient. So that was another way that we can, again, using magnetism, in order to create those that drug delivery system, and then now to show the way to those magnetic nanoparticles to go to a specific space or place in our body. Again, you may say yes, but again, does it work for deep organs? No, it doesn't. But again, we know the limitations of nanotechnology. However, we have the technology, and we just need new ideas in order to find different ways to do diagnostics, but of course to do different treatments. So magnetic nanoparticles are also, again, very used. I mean, they are used in medicine as well. Now, another use of nanoparticles is in disease detection. So here, of course, on the right, you can see the a picture. On the top, it's the baseline. And then after, again, some time, we used some nanoparticles. And at the bottom of the picture, you can see that now we see the surface of the cells that are, again, affected by a disease condition. Now, we can use the nanoparticles for cancer and for virus detections. We can use carbon nanotubes. We can use gold nanoparticles and nanodots. You will also hear about nanowires. These are all different nanoparticles that are used for the disease detection. We can also use silicon nanowires for gene detection. So for any type of gene that is causing a disease condition, and then we can use nanowires in order to detect if those genes exist in a tissue or in a cell. It's very interesting to see, again, how we can use the nanotechnology in medicine. Now, for the cancer or virus detection, for example, on the right side, you can see again a picture with again different components that I'm going to explain. So here we talk about the carbon nanotubes. They are covered with monoclonal antibodies. So the structures Y that you can see are monoclonal antibodies. So as you see, we don't make them, we can produce them again, in using existing, for example, cells. And then once we have those monoclonal antibodies, we are going to put that on a surface. Now, antibodies for growth factor receptor are commonly found in cancer cells. So what we do is that we are going to use those antibodies, and then once we again use those antibodies, we are now going to again add some type of cell preparation in order to see whether or not we have again growth factor receptors. These are receptors that we have in the cancer cells. So by using these antibodies that are our nanodetectors, we will be able to find out that yes, preparation from this tissue is showing that that tissue has a higher level of growth factor receptors. Hence, these cells are now again prone, or they are already having a cancer. Again, obviously we have 100 types of cancer, but here is just one example of again of a way that you can detect cancer. We can also use silicone nanowires. They are similar in use to nanotubes. We use antibodies that are attached to the wire. Okay, so these are the nanowires. These are wires structured like wires. They are very small, and we are going to attach again antibodies into those wires. And then we are now going to again use those nanowires. We're going to put them in contact with the cells, and same thing with the same principle. We are going to see if those again cells, they are having growth factor receptors. But this time, as you see, we are using another nano test, I would say, or nano detection. Now, another, I would say, system that we can use is gold nanoparticles and nano dots. So, similar application for a cancer and virus detection. We are again using or having antibodies attached to nanoparticles. You see, their antibodies are the vehicles for nanoparticles. Now we have nanoparticle antibodies that bind to cancer cells. So by having those antibodies binding to cancer cells, we are going to see the accumulation of gold nanoparticles on the surface of the cancer cells. Now, when we are going to, again, use a light, we'll be able to see these, again, the cells, only the cancer cells as well. Again, now, of course, we talk about shapes and size that can affect the color. But again, you may say, now, if you would like to use this, again, in vivo, in this case, or do we have any limitation? Yes, we do have again limitations, because you may want, you're now going to be able to see the gold particles or nanoparticles on a cancer cell. However, again, if these are in the deep organs, still we need to be able to use the light, again, which is not always easy. But at the same time, we also need to, again, I would say, expose those cells to the gold nanoparticles. But I can tell you that we can make biopsies, and we can use the gold nanoparticles and nanodots in the cells that are, again, that we have in our cell preparations as well. So not all of these diagnostic tests can be used in vivo, but again, we can use them in order to look into whether or not we have cancer cells or virus infected cells. Now, another interesting use of nanoparticles is gene detection. Here we use wires, I should say nanowires. So these are silicone nanowires, so nanowires made of silicone. They can detect specific genes. Now, what we do is that we are going to decide which genes, and based on the genes that we would like to detect, we are going to choose the sequence of nucleic acids that we will attach to the nanowires. So you see they are again very small tools. These are again nano tools that we are using. Now after having created the specific sequences of the nucleic acids, we can attach them to the nanowire, and then we will be able to use it. So we have sensor capable of differentiating mutated and non-mutated genes. In this case, we don't need PCRs, and of course this is going to take less time. So again, here we can use this in the labs as well. And now we can also use again nanotechnology in imaging techniques. So here we can use, of course, conventional techniques with X-ray, MRI, fluoroscopy, or CAT scan. We can use nanoparticles that we can inhale, and then they are again based on their biological and physico-chemical and biological properties. They can now attach to specific type of, for example, cancers in the lungs, like here what we see. And then we'll be able to visualize or to see those nanoparticles that are attached to different specific cells, like cancer cells. Now again, limitations, as always, we have again benefits, but also limitations. Is the limited detail. It means that when we are using nanoparticles for this type of imagery, we may not be able to really have a high resolution. We can just look into, again, what you see here. So it's just a cloud of cells based on, again, what we use as a nanoparticle. And of course, it's not easy to track the movements. That's because when they are adsorption, the adsorption is going to cause, of course, these nanoparticles to stay in place. So they are not going to move obviously in all of the cases when we use them. Now, for molecular tracking, for example, we have another type of nanomolecules. We call them quantum dots. Now, these are dots attached to molecules before injecting them into the, again, the body of the patient, for example. We can also use fluoroscopy to track the movements. These are colors from dots seen and imaged. Now let's go over these quantum dots to see what they are exactly. So of course we'll see this in the next slides for the quantum dots. Now let's go over tracking the blood flow. So here we can of course tag proteins of cells with gold nanoparticles as well. Again, the view is a process of looking, for example, at angiogenesis or the production of new blood vessels. So this is again another property of cancer cells, where they are having a higher number of receptors for growth factors, as well as their ability to, to provoke angiogenesis, because as you know, these tumors, these cancer tumors, they need to divide, they need energy, they need more blood, so they also cause angiogenesis. So we can use, of course, nanotechnology for cancer imaging as well. So we can inject gold nanoparticles. We can also will be able to see the localization around the tumors, and we can also use CT scan to show cancerous regions. Now let's go over the potential application of nanoparticles in medicine, of course with more emphasis on the cancer diagnosis and therapy. So let's talk about the nanomedicine, what it is going to bring to us. So these are nanometer-sized particles that have optical, magnetic, chemical, and structural properties that set them apart from bulk solids, with potential applications in medicine. So we can use them in drug delivery, as we see. We can use them in medical imaging. We can use them in therapeutic, ways, or the different therapeutic, ways of different therapeutic strategies mainly, and then we can also talk about diagnosis and sensing. Now, let's look into the facts about nanomedicine. On the left side, you have a graph. This is related to the interest in the area of growth for the use of nanomedicine. So we see that we were looking into from 1990, and we see that after year 2000, we have an exponential increase in the number of patents. So these are dots or rectangles in blue. And then you also have a very interesting increase in the trend of the publications. This is the line with dots in red. When you look into the drug delivery in the different areas, this is what we see on the right side of the slide. You see that the majority of the use of nanomedicine is in drug delivery. But we also have, which is also large use, this is in vitro diagnostics. And obviously it's going to be followed by biomaterials, imaging, drugs and therapies, and active implants. So mainly for now, we are looking into in vitro diagnostics and drug delivery. Now here we can see the drug delivery in, again, different technologies in nanomedicine markets. So here on the left side of the table, we see the healthcare sector, and then we have the number of products, followed by the sales in billions of dollars, and then the total advanced stage and different number of companies. So what we can see is that, for example, for the drug delivery, we have 23 medicine products for the sales of $5.4 billion. In total, in the pipeline, we have 98 products for delivery drugs. And then, of course, we have about 113 companies working on drug delivery systems using nanotechnology. And of course, this is as of, year 2006. I didn't find any other article exactly going over this type of data, but obviously we can find similar data for the recent years as well. But as I already mentioned, this is really increasing in an exponential way. Now coming back to the drug delivery, because of the small size of nanoparticles, they are taken by cells where large particles would be excluded or cleared from the body. So all we need is just to have a system, a nano system, where we will be able to have a structure usually surrounding that nanoparticle that will be recognized by our cells, so that we can use a normal biological activity, which is endocytosis, so that that nanoparticle or nanosystem will penetrate the cell. Now what happens then is that we are looking into making a system, a nanosystem, where once it undergoes endocytosis, it will also be able to release its content. Now the content may be a dye, maybe again a molecule that is going to emit some wavelength, it's going to emit some light, and then we can see that specific cells are, for example, are having cancer. Or it may be the content may be a drug, it may be something to stimulate a kinase or inactivate a kinase. It can be anything, but we can use them as a drug delivery system. So I'm going to again read, of course, the explanations for the endocytosis that we see here. So for number one, or again the first step here, is a nanoparticle carries the pharmaceutical agent inside its core, while its shell is functionalized with a binding agent. Now you see very well that we can have different binding agents. The binding agent, of course, it can be, again, any molecule, biologically active molecule that we can make or we can extract from different cells. We can also have cell cultures, and we can create those molecules that we can attach outside of the, again, a bilayer membrane that we can also make artificially that will surround our nanoparticle. We can do this in the lab, obviously. Now, in step number 2 is through the binding agent, the targeted nanoparticle recognizes the target cell. Now, the functionalized nanoparticle shell interacts with the cell membrane. So at the end of the day, we are looking into finding ways that selectively our nanoparticles are going to adsorb to the a cell with a disease, and then they will be able to penetrate through the endocytosis. So step number 3 here is the nanoparticle is ingested, okay, this endocytosis, inside the cell and interacts with the biomolecules inside the cell. Now again, what is in the nanoparticle? In step number 4, the nanoparticle, nanoparticle particles breaks and the pharmaceutical agent is released. Now the pharmaceutical agent can be, again, a dye, can be just a color, it can be again a phosphorescent product. It can also be, as you see, a pharmaceutical agent. So this is what we are looking at when we talk about drug delivery as well. Now, talking about the drug delivery, nanoparticles for drug delivery can be metal, they can be polymers, they can, or of course, lipid-based. So below what we can see, these are examples of, again, the nanoparticles. As you see on the left side, you have the, again, the small interfering RNA, siRNA. Okay, this is a double-stranded RNA molecule that can, for example, silence genes. And this is within a micelle or within a bilayer membrane that we can prepare in the lab. And on this, membrane, artificial membrane, we can now attach antibodies that will recognize specifically different tissues. Now, those tissues may be cancer cells, having cancer cells as well. Now, this can be, again, used in vaccines, okay? We have not small interfering RNA, but we may have RNA vaccine, based vaccines as well. They may also have this type of micelles or the lipid bilayer, and the mRNA is going also to penetrate, I would say, different tissues as well and to do their work. Now, what you can see is that when we are using the small interfering RNA, so in this case, we can see on the picture on the very right side of the slide that you have sick tissues that are treated with a targeted nanoparticle. So when we are using antibodies on the nanoparticles or for a drug delivery nanoparticle, we can now make sure that those antibodies are going to conduct the, the drug in the nanoparticle directly to the sick tissues, because we already know, or we already made the antibody for being attracted and to, be adsorbed to the sick cells. If not, if we were not using, these non-targeted nanoparticles, we would see in the middle, as you can see of the slide, we can see sick tissue treated with non-targeted nanoparticles. So by using those nanoparticles, we can create, drugs that are very selective in a way that they are going to be able to approach and to adsorb to the cells with a disease, and then they are only going to, again, target the cells with a specific disease. Now, let's go back, as I already mentioned, let's go back to quantum dots. The quantum dots are used in medical imaging, and they are, again, nanoparticles as well. Now, optical properties of nanoparticles depend greatly on its structure. Now, particularly the color, which is a wavelength that we perceive with our eyes, emitted by a quantum dot or QD, depends on its diameter. So on the left side of the slide, you can see the quantum dot or QD structure. Depending on its diameter, it's going to give you a different wavelength. It's very interesting because now we can use these quantum dots at several sizes or diameters. We know now that different diameters are going to give us different colors. So it allows us to look into specific structures on a cell or in a tissue where we are looking into, again, for example, maybe the rate of penetration through a channel of some type of ions. Or we are going to look into the size of, again, the molecules that can go or pass through a channel or ionic channel. Here we can, for example, expose those cells with a specific channel, ionic channel, and then to use different, I would say, quantum dots at different diameters, and then we will see outside, of course, of the cell that we have different colors or a mixture of colors, but inside the cell we will only see one or two colors. It shows that the diameter of the molecules or the ions, for example, that can pass through those channels is what? Again, based on the color that we can perceive, which is, again, depending on the diameter of the quantum dots. So it's, again, another very interesting tool, which is a nanoparticle as well. Now here we can see the quantum dots that can be injected into a subject and then be detected by exciting them to emit light. Okay, so you can see again now in different structures here. These are imaging of quantum dots, again, targeted on cellular structures. Okay, this is again an article that you can find in Nano letters. Okay, it's already at the bottom of this picture, but again, this is just in 2008. Nowadays, we really made so much progress, and of course, we use also those quantum dots in several medical imaging strategies. Now, coming back again to quantum dots, because this is very interesting, the quantum dot itself is toxic, of course, so therefore some typical modifications must be made for it to become biocompatible. So what you can see, first of all, is the core. So number one, on the left side, you see the body or the core of those of the quantum dots or nanoparticles. The core consists of the semiconductor material that emits light. This is the quantum dot that we talk about. Then around that, and we can do this in a lab, there is a shell that consists of an insulator material that protects the light emitting properties of the quantum dot in the upcoming functionalization. And then you have in again, number three, this is in natural coating. So the shell is functionalized with a biocompatible material such as PEG, which is a lipid layer. We can also make lipid bilayers in a lab. It's something very, very simple to do. And again, by doing this, of course, we can also reconstruct, again, a membrane that is not exactly the same, but similar to the cell membranes as well. And of course, we have to add on this the antibodies that we also create. And those antibodies are extremely selective for specific proteins that are on the surface of target cells. And then number four is additional functionalization can be done with several purposes. So here we can embed a drug for drug delivery. Here we can add an antibody to become the quantum dot target specific. So this is a system here in front of you. There's a system where you have an agent or a nanoparticle with some, I would say, function. So here is just emitting light. You also have a shell, which is an insulator. You also have a membrane, but here we talk about an external shell here, like a natural coating. And over that, you have now selective immunoglobulins or antibodies, or we can also attach to these antibodies and enzymes or antibodies and vesicles containing some drug, and then we will be able now to see where those quantum dots are going based on the antibody that we did select and made. We are going to again, we can see where those quantum dots are going to be absorbed, and then where they are going to undergo endocytosis, and where they will be able to emit some light, so that we will see that those quantum dots are now within a target cell. And this allows us to see that since those quantum dots are in the cytoplasm, the drug that we wanted to deliver to that, again, a cell is also in the cytoplasm. Now imagine how, and again this is what's happening now, how we can really make so many different products, so nanoparticles that will be useful for us, because we'll be able to engineer them to recognize specific cells. We are going to have hundreds of thousands maybe of target cells, where we are going to have those quantum dots that allow us to not only deliver the drug to the specific cells or the tissue, but at the same time we can see that it did its job because it also emits light. So very, very interesting use of nanotechnology. And here, of course, we can talk about a nano system. Now here, this is again what I was just explaining in the previous slide. So using a drug delivery-like mechanism, a targeted lipid-based nanoparticle, so here we use the abbreviation TNP in different publications, encapsulating quantum dots, specifically attacks a cell having the receptors that pair with its ligand coating. Of course, we talk about the antibodies as well. So upon ingestion, so here we talk about endocytosis and destruction of the targeted lipid-based nanoparticle, the quantum dots are set free and accumulate in the intracellular structures or on the intracellular structures. So this is again what we can expect from those nanosystems. Now let's talk about the future on nanotechnology in medicine. On the left side of the slide, you can see, of course, in the middle, those different nanosystems that we already explained. Now we can use those for performing nano implants like stem cells that we may now attach to our nanosystems, and they will be directed to a specific site in order to bring stem cells within the site without a need of any invasive, I would say, any surgery. We can also perform disease diagnosis. So these are for devices and for labeling. We can also do early detection of different diseases like cancer. So here we talk about molecular imaging. We can do monitoring through the biomarker mapping. We can, as I already mentioned as well, have a targeted therapy, like for example for cancer, but not just the cancer, also viruses. We can also find therapies for flu, for example, having again these nano systems in order to recognize and of course selectively and to attack flu I would say, cells that are infected by the flu virus. We can also have theranostics. This means to see and to treat any type of disease condition. We can have drug screening. We can also do what we call gene delivery. You know that in gene therapy, which is again not ethical obviously, but we are talking about the science itself without any practice. The gene therapy is again based on again insertion of the genes or repairing the existing genes, but inserting of genes in the DNA so that patients will be able to get new genes that are having or they're expressing naturally, and they are going to give again a product that if we don't have the product, it would now again cause a disease condition. But the challenge that we have is not to make those genes. The challenge is how to insert those genes or the new genes or repair genes in the DNA at the right place. Because if we insert a gene anywhere in the cell, and if you don't control it, and this is what happened in 1980s and 1980 and 1981, when we had unfortunately 2 victims in 2 different countries, in France and England as well, those who unfortunately they passed away after a week of treatment with gene therapy. That's because we were able to insert genes, but we were not able to insert them in the right place. Now imagine that a gene is inserted in a for example, a region for a promoter region. It can be in the middle of another gene. It can be nowhere in any, I would say, sequence of the DNA where there is no expression of the genes, for example. So it may cause risks. It may cause cancer. Again, as I told you, we had several victims of gene therapy because we couldn't control where those genes were going to be inserted. By using nanotechnology, obviously we can see some promises. We may see that, yes, we may use nanotechnology in gene therapy in order to deliver the gene at the right place in the DNA. But again, gene therapy is not ethical. But I'm talking about the science behind and the knowledge about the gene therapy. We can also, of course, do the tissue engineering. Now, the same thing with stem cells, I already told you. We can, related to nano implants, we can just look into not one type but several types of stem cells. And we may also now, for example, create some implants, nano implants, and then we are going to look into maybe reshaping or making a new tissue. Okay, so again, these are all promises, they are all plans, these are all possibilities. You can call it the way you would like to, because again it will depend how we use nanotechnology. I talked about the ethics maybe more than 20 times in this lecture, because ethics is not only taking care of the well-being of patients, but it's also using the instruments and the science that we have in a way that we are not going to harm anyone or even their, I would say, their descendants as well. No, don't forget that when we are looking into gene delivery as well, we may change or affect the descendants or the offspring of animals, or again now the future of humans and their families as well. So these are all ethical considerations. Now you may say we may also go over the informed consent. Obviously, we can always say to our patients that yes, this is an experimental drug or delivery system, and we may also talk about risks and benefits. But still, if you are dealing with the genes, with different therapies, with using stem cells, this is not just a matter of giving a molecule and then the molecule is going to be metabolized, and then again it may have sometimes some toxicity level as well. We can adjust the dose or looking into removing some impurities, for example, but anyways making that drug safer. But if you are affecting the genes now, if you are affecting, you're now creating again a new tissue, if you are having nano implants, it may not be as easy as just taking again a drug that will be metabolized, and you may not see that in your body after an hour or after a day. So ethics is again, this is another very important, I would say, topic when we are talking about new therapies in medicine. Now, let's go over the schematic diagram of the current trends of micro and nano robotics in precision medicine. Why? That's because when I was talking about the use in medicine, I should have used the word precision medicine, meaning that we are not looking into targeting cells, looking into precision of our interventions. And when we talk about the precision, we can now look into, again, different use of nanotechnology. So here we talk about nano robots in the lab and, of course, in clinic. Now, for the lab, for example, we can look into, you can see on the top left side of this, again, picture that you have on the slide, for the delivery of the drugs. So here we can look into the delivery of pharmaceuticals, biologics, and delivery of genes, if ethically acceptable, and again, it's safe. Also delivery of living cells and inorganic therapeutics. We can also use, again, nanorobots in precision medicine in imaging. So here we also went over a few examples for, again, using them in the optical or in ultrasound or magnetic or radio nuclide, I would say, imaging. Now, we can also use nanorobots in precision medicine in clinic. So here we can use them for biopsy, in surgery, of course, in biopsy, in sampling, in tissue penetration, intracellular delivery, and biofilm degradations. We can also use them in diagnosis. So we can use them as biosensors for, of course, isolation of different cells, and as physical sensors. Now, what are the challenges when we are talking about nanotechnology in medicine? We talk about real-time monitoring of drugs distribution, action mechanism, and patient response. Indeed, this is a challenge because when we are using nanotechnology, we may not be necessarily able to do the real-time monitoring of drug distribution or action mechanisms. So this is one challenge. Then we have another challenge related to the fast detection of biomarkers at lower limits. That's because, again, it cannot replace for now, at least, when we are doing a detection of biomarkers by just to drawing some blood sample and then to measure that. But still, it has some, I would say, chances to be obviously improved and then to be used. But for now, these are challenges. Another one is understanding the mechanism of diseases. So we cannot use for now, of course, nanoparticles. This is limited, okay? You cannot make a nano camera and then to again have it implanted in a cell or in a tissue, and then having the camera observing, okay, how again the cancer, what's the mechanism of disease like cancer, okay? We cannot do that. So there's another challenge. Then we have another challenge, which is diagnosis leading to personalized treatment. Now here what happens is that we are using, of course, now technology for different diagnoses. However, from doing diagnosis into developing personalized medicine, we have a challenge, and the challenge is the cost. Because nowadays, of course, we are not, using that much nanotechnology. We use it, of course, when the technology used is well established, and it's also fine-tuned and then improved, and then it becomes cost-effective. If not, if you would like to go toward the personalized treatments, obviously this is the dream for everyone. To me, this is exactly what we should do. But again, we have challenges, and some challenges, majority of challenges are, of course, here, the cost as well. But we have the technology. Then another issue or challenge is the detection of deep tumors, not just the tumors, but any organ which is deep. And if we cannot, for example, use even the imaging, for example, for the imaging, we also talked about, again, those quantum dots. If the organ is deep, obviously, even if the quantum dots are going to penetrate the cells, we will not be able to, again, emit to them some light and then to, I would say, collect the light or to even be able to see the light. So we cannot really use those nano systems in deep tumors or deep organs. Another one is selective targeting is in extremely heterogeneous tissues. So here, same thing with the cancer cells, for example. We know very well that in cancer tumors, we do have several types of cells. We even have sometimes stem cells as well, because as you know, the cell is, cancer cells in different phases, of course, they are losing their ability to control their division, obviously. And we also see that there are different, I would say, cells that are going to be made in a tumor. So when we are using nanotechnology, another challenge for us is to really deal with heterogeneous tissues. But still, even despite these challenges, nanotechnology is very promising for the future. And obviously, and I already explained at the beginning of this lecture, these nanotechnology products, as you can see, are medical devices and combination therapies. Now, when we are looking into the registration of these products, obviously, we need to know more or learn enough related to, I would say, about the products as well, their own mechanism of action, their physical, chemical, and biological properties. And at the end of the day, don't forget that ethics is primordial. It's very important because, as I already explained, we have these tools. We are working on having more tools, more selective tools, safer tools as well. I'm talking about nanotechnology tools. However, we always have to take care of the ethics, because some manipulations, some treatments, if they are not controlled, some gene manipulations, if they are not controlled, they can really cause huge harm to the population. So thank you so much for joining me for today's lecture. I hope that you enjoyed, again, this presentation, and I wish you all a very good day. Thank you so much.